So I'm going to be talking about prokaryotic DNA. This is what it looks like as a whole, and then this is what it looks like in further detail about replication. So for my example, you're going to be looking at the origin of replication being on your right, and then the direction of replication going towards the left. So in further detail, this is the prokaryotic replication fork, and replication occurs at the origin of replication, where there are about 245 corresponding base pairs along it. With the sequence of base pairs, different proteins can recognize their binding site. It occurs in the 5' to 3' direction, resulting in leading and lagging strands. Initially, helicase B unwinds the DNA, breaking down the hydrogen bonds between the nitrogenous pairs. Gyrase, which is A, additionally helps helicase unwind the DNA as it falls within the class of topoisomerase and continues to relieve the strain on DNA. However, there needs to be another form of separation, so to keep the strands separated and stabilized, ultimately, just to prevent the double helix from retracting, single-stranded binding proteins, which is C, bind to them. Before replication can occur or a new chain of DNA to emerge, primase, which is H, acts as an RNA primer that synthesizes the 5' ends. Primase allows for DNA polymerase 3, which is D, to do its job, which is to synthesize new strands by polymerizing nucleotides, using the parent strand as a template. So it stays along the parent strand, which is in the direction of 3' to 5', and as it goes along, synthesizes the new 5' to 3' strand, breaking down 3 phosphate groups. In other words, nucleotides are the energy that DNA polymerase 3 needs to continue synthesis, as it actually uses 2 of the phosphates. When DNA polymerase 3 attaches to the primer and adds the nucleotides until it reaches the next RNA primer, then DNA polymerase 1, which is E, removes the RNA primer and replaces it with DNA. Lastly, ligase, which is F, seals the new strand by repairing the remaining nicks in the DNA. This enzyme seals the broken phosphodiester bond, and Okazaki fragments, which is G, are the small pieces of DNA that are synthesized discontinuously. However, ligase is actually the enzyme that links them together to create the lagging strand.